That's a load of science.
Hello, everyone, and welcome to another episode of That's a Lot of Science. Today with me, it's Carlo Barbera, project manager at GSK, and we are talking about a very interesting topic today. So the drug discovery process on its own, but also like the management of and how to communicate about drug discovery in the outside world. So first of all, Carlo, thank you very much for being with us today. And as we always start like this, I would like you to give us a little bit of an introduction of yourself, what you do, what's your background. So I leave the stage to you.
Thanks, Alessio. Well, first of all, thank you very much for inviting me and having me here. It's a great pleasure, and I always like to talk about science and about drug discovery. So yeah, really appreciate that. I'm a scientist by training, so I've got a master's degree in biotechnology, and then I got a PhD in biomedicine. And for the last 12 years now, I've been working for the pharmaceutical industry as a scientist first in the preclinical science and discovery, and then as a project manager lately for the last 3-4 years now, different companies, different therapeutic areas, but always interesting science. So that's my background, and again, very happy to be here today, and I can't wait to dig into our topic of the day.
Thank you. So I mean, let's start on that then. So drug discovery. I mean, I've been working on drug discovery as well, so I have that background too, and it feels like from the outside that drug discovery is just one big thing that people don't really understand what's behind it. Let's say what's behind the curtain. So, I mean, with you and with your experience, I would like to go there into more details and how like new medicine are discovered and how they progress through all the preclinical phases into an actual medicine. So, like, I mean, if you can, give us a little bit like what happening step by step, and then we keep talking about this.
Absolutely. Like you said, research and development of medicine is really the core of the pharmaceutical industry. It's the oil of the engine, like we say. But also, it's a very complicated topic. It's complicated for us who work into the business, and it's even more complicated for people watching from outside. So I'm very happy to have the opportunity to talk about this, because I think we never speak enough about how medicine are discovered, how medicine are brought to the people, and this is very important to understand, and also maybe to break down some let's call myths about the pharmaceutical industry. So let's start from the beginning. So you said, how do we research and develop a new drug? First thing first, we need a target, right? So we need a biological entity like a protein, a gene, or also another nucleic acid that can be targeted for a drug. And this is the very beginning of the process, and it's called target identification. We identify a target that needs to play a role in some disease, and that's where all starts. Actually, this can take as little as days, months, or years, depending whether the target is already known. So we can start from maybe the literature, and if there's already a body of literature around a target or a possible disease mechanism, or we can start from other means, like in silico, so use computer models to better understand the science behind a protein, a gene, RNA, or any other biological entity that really sits behind a pathogenic mechanism. Or we can also do some wet lab already. So maybe biomarkers or proteomics, other omics science that can help us really understand better. So now that we have identified a target, and again, can be a matter of days or years, we can really get a little bit started, and we need to validate that target. So at this point, we really want to make sure that that target plays an important role in the pathogenesis or in the disease that we want to tackle. And so this is where again, there's a lot of work going on here for this phase. And again, it's very variable because pharmaceutical company tends to have standard timelines, or at least we wish we have a benchmark for the duration of the phases. And the sooner you get, so target identification, target validation, the more murky and smoky this duration is. So again, target validation is a very labor intensive phase, and it's really can get very complicated. But at some point you
get it.
you make it through. And
but is it something that you would say this is happening more in the academia, or the company invest also in these kind of things, like the very early stage? How does it work? You feel more like this is something, because the way you describe it is both like biological, so you have pharmacological people, like people that have biology background, molecular biology background, but also computational chemistry background. So you need a lot of efforts, let's say, and resources there. So how is this approached in a company?
Absolutely. This is a great question. I think it's a little bit of both. So that's probably where also you want to have some academic collaborations, because the earlier you get into the pipeline, so target identification, target validation, the more it resembles basic research. So yes, of course, we have in the company means and instruments to run those phases, but it's even true that some of the part of the science is not under our capabilities. So we really want to tackle this part and get the best out of collaborations with universities or something that maybe it's already known. During target validation, we also want to answer the question: is the target that we identified, can we get to the target with a molecule or something? Is that druggable? So once I get there, can my molecule or my my therapeutic bind to this target and exert an effect? So there's a lot of question at this point. There are always questions, but at this point they're more theoretical or more basic questions. So that's why you want to do that in-house, because generally in-house you have lower costs and quicker turnaround time, but also you want to exploit the knowledge that is out there in the academia, universities, or other companies possibly. So
I mean, you introduce a very nice topic, which is the druggability of targets, because there are a lot of targets that seems very promising and seems very interesting for tackle a disease, but then it's not possible to design a drug against them or like that binds to them. How this has changed, like in your experience? Like, do you see that more targets that were deemed as non-druggable are now becoming druggable? Is there some technique that are used to evaluate that?
Well, absolutely yes. I mean that's part of the knowledge that is changing. And if you imagine, like 50 or 60 years ago, when the first antivirus came out, they at the time they thought, okay, there's nothing so toxic or so that has a great efficacy and affinity to kill a virus and not to kill a human being. So and then antivirus came out, and so it was really and for that there was a Nobel Prize also for Gertrude Elion, truly, and that was a pivotal moment. So even now there are probably targets that we think are not druggable today, but yeah, with the increase of the knowledge, the amount of knowledge and technology that is really hitting our business, we're in a position to say that you never know. Today it's not possible. Think also the improvement in oncology, when yes, still the cytotoxic therapy is still a gold standard, but then immunotherapy, now there are ADCs, so conjugate between an antibody and a cytotoxic payload. So things are changing at an incredible pace today. And you probably know that because you're also into the AI and those technologies that are also pushing on the accelerator. So never say never. Of course, druggability of a target is still a challenge, and it will always be, I think.
Yeah.
But there are more and more tools we can use. There are more and more technologies we can take advantage of to discover new targets, make sure those targets are druggable, make sure the drug that we make are more and more specific, less toxic, and more they get more efficacy. So these are
great. Yeah. And we still haven't started thinking about the molecule now. We are still in the target.
Yeah. So we said target identification, then target validation. We have a target now. It's validated, so it's time to really get started seriously and with the lead discovery phase.
But before moving forward, a word from our sponsor, IDM Insight. IDM Insight is an interactive intelligence platform that brings the latest pharmaceutical AI innovation to early drug discovery and clinical teams, pairing the latest technological advancements from AWS Cloud and cutting-edge science disciplines. Insight is able to save drug development and clinical teams time and money by simplifying your interactions with intelligence data and AI solution into one centralized platform. Check out the platform for yourself at idm-insight.com. That's idm-insight.com.
We really want to start the discovery. So what's really called discovery, it's a specific phase, which is where we want to go from a huge possibly number of molecule. If we talk about small molecule, but even if we talk about large molecules, pretty much the same, and identify the hits, the so-called hits. So you want to start from a very large libraries, tens, possibly hundreds of thousands of molecules, and start screening. You can use many tools to screen that. You have biochemical or cellular tools like the high-throughput screening. You can have oligonucleotide-based methods like the encoded library technologies or even affinity, so like affinity selection mass spectrometry. But the bottom line is that you want to go from lots of molecules to a pool. The quantification depends really on the technology, on the company, but you definitely want to come from like hundreds of thousands or tens of thousands to maybe some thousands at this point, and you have your hits. So a number of molecules, chemical series or clones of antibodies that you like somehow, because like we said, they possibly can bind the target, or they got an affinity for that, or they're involved in the pathogenesis, or anyway, it's something that you feel is something that can work.
Of course, these hits are not as well like very advanced molecules, so there is like room for improvement. I mean, you find molecules that are as well like early stage, so then you want to have that information, but you don't want to be as well too restrictive on the type of molecule, because then on that is based all your chemistry campaign, let's say, around this molecule, right?
Absolutely.
And that is where the medchem work starts.
Yeah, absolutely. Of course, you want to have some quality criteria to define, yes, or also the criteria through which you evaluate your stage gating. So you arrive at some point, a milestone in your project where you want to say, okay, I'm happy because I reached this and this and this criteria for my chemical series, for my molecules, for my product in development. So but absolutely, you want to leave also as much doors open as you can. Of course, you already start looking at properties like, I don't know, solubility of a molecule, possibly efficacy already, possibly, like you said, pharmacokinetics, so how the drug interacts with your body and how is absorbed, distributed, metabolized, and excreted by your body. But Absolutely. The goal of this phase is to identify the series that can then be optimized, because the next phase will be lead optimization. So at this point, you don't have hits anymore, you get leads. So an even smaller number of molecules that have even better properties, and you really want to start play around with substitute chemical moieties, or try to modify the properties, like we said, solubility, the absorption rate, any other things that maybe are not optimal with your chemical series. But yes, the lead optimization phase is really what's this for, to make sure you improve the properties of your molecules and you make a better molecule that really try to match your needs, because it's always a challenge.
It depends on the aim and the objective that you have. Yeah, of course. And all of this is mainly done by chemistry modification of the structure. So you have to synthesize. I mean, here we're talking about thousands of compounds, so that you have to iterate through this process. So synthesis, analysis, test. Then based on that, you evaluate the structure activity relationship, and then you go back in the lab and you synthesize other molecule. I mean, I've been there, and you have been there too. This is a process that first of all, I feel it's very it's very challenging in terms of course, like every molecule that you synthesize, you feel like that's the one, that's the best.
You hope so. Yeah, you definitely hope
most
so.
of the time it's not.
Yeah.
And also let's
let's talk a little bit also about time, because maybe people are interested in. So like we said, hit identification validation, it's a little bit murky, so can take days to years. So we don't talk about that too much, but let's say about lead discovery and lead optimization. So lead discovery, if you're good and lucky, because luck is always part of the game, 1.5 years should be enough to really get you going and pass from the heat to the leads. And you're at this point, you're also ready to go to the governance and achieve the milestone and go to the next phase, which, like we said, is lead optimization. Mm-hmm. At this point, lead optimization, you got at least 2 years of hard work and huge costs, because, like we said, the chemistry, if we're speaking about small molecule, will be the majority of the work, because we're going to try to optimize the series that we have and the series that we selected to improve, like we said, properties like solubility, permeability, efficacy. And of course, you will be working hand in hand with the pharmacokinetic and pharmacodynamic colleagues. You also want to start a little bit of development, because so far you're still in research. Now, it's very important. Research and development are two separate things, even though they always come together, like Laura and Hardy. So, and you want to start, also to ramp up with your chemical process for the synthesis that will be required to support the production of intermediates, or the API, so the pharmaceutical principle that will be necessary for further studies and to support also the DMPK and the efficacy studies that you're already carrying out. So 2 years, it's a good estimate. A little long, a little less. It's good, but yeah, that will be definitely a target for this phase, the lead optimization. At that point, somewhere in the lead optimization, you will have a pre-candidate. So a molecule that you really like and you think can be the one, and you want to start determining the human dose prediction and the safety profile of that molecule using like dose range finding studies in rodents or other animal species. Mm-hmm. And if you like what you see, you're ready to make the all-in and say, this is the one. This is our candidate, okay? This is the molecule that we're bringing into the development.
Yes.
So
the development is, when you move from like targets, like isolated targets or cell study or animal study towards the human, let's say, study. It's more like when you start looking at the actual use of the medicine.
Again, different companies have different way to see that, of course, but traditionally, the development starts when you have a candidate. So you move from thousands of molecules to one, to really one, and that's the important part, because you want to start develop the process for producing that at the industrial level. You want to start the studies, the preclinical phase. So the animal studies that would be required to for registration. So to have the to go to the authority and say, look, this is safe, and those studies will be also necessary to go the first time in human. So you want to make sure the molecule is safe in the animal before you test them in human beings, in people. So that's where traditionally we see the end of the research, which is more let's say, a try and error phase compared to the development, which is hard, but must be doable and something you can and programmable. So you need, it's an industrial process. So you need to have the technology to do that, and you need to plan in advance everything to make sure you achieve your development steps.
And also in this phase, you start thinking about the formulation of the drug as well, how this is going to be administered, how this is going to be, I mean, which formulation is better, which improve as you said already, the ADME properties, absorption, distribution, metabolism, and excretion. So you start already thinking about this not only as a molecule, but as a medicine.
Absolutely.
And also, like you said that before, like the research part, it's there is a lot of overlap. It's a iterative process, so you can go, it's try and error, so you can go back and forth a little bit. Once you select your candidate, it's a little bit, you close that door a little bit. No, now it's only ahead.
Yeah, you hit the spot. It's all about that. So that's why you want to make sure it's the best molecule, and somehow you never know until you get there. But yes, you close the door at that point. You chose that molecule. You made all in on that molecule. That is the one that we want to bring forward. That is the one, because so far, okay, you can get back, like you said, iterate again and say, okay, we can change the molecule. But once you start the development, once you start the registration studies in animals, you cannot change a lot. Things are very constrained, because the pharmaceutical industry is the most regulated industry on the planet. And when you go for the file for the registration, the authorities will want to see how you develop that molecule, what kind of studies you perform, and what kind of processes are you have in place to produce that molecule, the chemistry, the manufacturing, the control. And you cannot just say, okay, we did this for the first study and then we changed completely the process because we wanted to do another molecule slightly different, but it's pretty much the same. No, nobody would like that. So you got to make sure consistency of your product and the consistency of the process that you use to show the safety and the efficacy of that product.
Yeah, from the registration part then. So it is this is the first step for the registration, because like usually you protect the molecule from a patent perspective. You can go for a patent, but you do that probably during the lead optimization phase. But in that phase, you don't have to provide too much about safety. It's more about this molecule can be potent, and we go with it. Now it's a all around description of your molecule. So how that change also in terms of investment, because a patent costs money, but probably for a company it's not a big investment to have many patents. They prefer probably to have more, but for the candidate, probably the budget and the investment needed and the time needed is different. So how is the approach there in a company, like when they decide to go forward?
Yeah. Okay, from the patent perspective, you're right. Even though you won't find two people who have the same opinion on when to file a patent for a molecule, let's say the lead optimization is probably a good point, where you already have an idea of what your molecule will be, and therefore before declaring the candidate, you can patent that anywhere from there on. Not too early, but not even too late, because you don't want to be scooped by anybody, and there's a lot of competition in every field. Even though you're doing something maybe new, but there will always be some other people doing similar things. From the safety point of view, let's say that safety is always your concern, because more than efficacy, you want to do something that is safe. Maybe that won't work like you thought, but the most important thing is safety, because when you go into human, it's okay to fail because your molecule doesn't work, but you don't want to fail because you harmed people with something that is not unknown. So it's never too early to investigate the safety, even into the lead optimization phase. And your candidate, you already have a very sound idea of the safety. So you have, throughout the research part of the life cycle, you have target safety review to make sure, yes, you don't have cross-reactivity of your molecule with other, you're specific to your target, your molecule is not going somewhere else to make damages. And the preclinical part is really when you want to show that in a control environment. So the big difference is that when you're in research, lead optimization or lead discovery, the safety that you do is non-GLP, so is not carried out under good laboratory practices, which means like an extreme standardization of the procedures in the laboratory, how animals are treated, how your product is treated. When you go for the registration studies, so those studies, you want to go GLP. So you want to make sure those are carried out under good laboratory practices, to make sure you show the authority. You did everything in your power to control the results and make sure these are as reproducible, as standardized as possible.
Yes. And this is something that exactly. I mean, in the phase that I have experience with, you don't really care. But is there like something that even in the early stage we can do in order to, let's say, make life easier for the people that works after this like lead optimization phase, that works in the development phase, to help them to not have to rework or restart from almost the beginning when they decide the candidate?
Again, this is a very, very interesting point. Consider the molecule that is going through the different stages of research and development like a child. Okay, research is a young child going to the elementary school and probably the middle school, but then you go to high school and the university, and if you didn't have a solid background, and if you didn't do your homework when you were at the lower schools, you won't do good at the high school or
No.
university, right? It's pretty much the same. So if the research does not work well, let's say, or at least if they don't do the design of the molecule and of the product, because it's not only the molecule, it's a product, like you said, formulation and a product. If you don't do a good design, you're going to have problems during the development. So you won't be able to scale up to big volumes, or it will be very expensive to do that. You're going to have problems of formulation. You're going to have problems of precipitation, for instance. So it's really it's a really difficult, because somehow it's like those parts are disconnected, but they are not. One flows into the other, and therefore it's very important to do the research, which is already difficult itself, bearing in mind what will come next. Otherwise, if you just work separately and you just take those phases separately, it's going to be very difficult to develop a product. You will incur into problems that will ultimately bring the project to failure.
Yeah, exactly. So you have always to keep in mind that, the part that, because we usually work on only one part, like as a scientist, you work on maybe on, so you are either like a lead optimization scientist or like a, development scientist, but you have to already, since the very early phases, you have to think about that as a whole, as a process where everyone brings like a brick towards the building of the house, and the house is the end of the is the end product, is the drug and the medicine. And if you put your brick wrong, then the whole house will be a little bit wacky.
Yeah, unsteady,
and
to say
nobody
the
will
least.
want to live in there.
Absolutely.
and
Absolutely.
and yeah, as you said here, like now it becomes more of an industrial process. So you have to, scale up the synthesis. We're talking about, kilos of materials. We're talking about, setting up maybe also a production line for the formulation. And then what happened? How we start the clinical part? So where we decide, okay, now we are ready. I mean,
Yeah. So we have the candidate. We go into the preclinical phase, which is driven mainly by, like we said, the safety studies for the first time in human studies. But also chemistry, manufacturing, control is very important in this phase, because we want to produce the drug substance, and which is just the API, the active pharmaceutical ingredient, and we want to produce the drug product, which is the formulation that can be, I don't know, something you can take orally or an injection, but the final product. And we also want to produce the clinical trial material. So the product that will be used in the clinical phase. And if we're, again, good, but also lucky enough, can take, let's say, 15 to 20 months to close the preclinical phase, or at least not close the preclinical phase, because the preclinical phase and the safety study will go on through phase 1, phase 2, and phase 3 of the human studies, but at least have the minimum requirements to go into human. So I think that's a good approximation, 15 to 20 months, if you're, again, good and lucky. So at this point, I mean, we can really shake hands because we are at the point where we are ready to go into human. And to do that, we need a regulatory approval. You cannot just decide we run a clinical trial. The authorities must be aware, and you need to obtain a so-called investigational new drug, which is called an IND in the US, or a clinical trial application, also called CTA in Europe. This is your pass for the clinical phase, because to move a non-approved drug within a country, you need a permit, and this is all about. And also to test that into humans, you need a permit. So you want to, and like we said, it's all about showing your drug is safe. And it's being produced under the most stringent and best standards of the industry. That's all about. And then once you're into human, congratulations, because now you've got 10% probability to reach the approval. So let's say the big job is done.
Yeah, exactly. Everyone thinks like, okay, now we are in human, we are done. But no, I mean, there's still a lot ahead. And,
jokes aside, jokes aside, if you look at the statistics, and you can Google just how long does it take to develop a drug, or what is the likelihood of approval, you always find 10%, and this comes from only the clinical phase. So there is a higher percentage of passing the phase 1. Phase 1 is where you have a smaller number of patients that you're treating, generally tens of patients, less than 100, and you want to test specifically the safety. So you can also take a look maybe at the efficacy, but it's not the primary goal of this phase. The primary goal is to make sure your drug is safe in patients that maybe, if it's not a life-saving drug, can also be healthy patients. And therefore, at this point, you have a pretty high, at least statistics say that, a pretty high probability of success of going to phase 2, which is around 65%. So 65%, these are the stats. 65% of molecules that go into phase 1 make it to phase 2. Phase 2 is the death of valley, sorry, the valley of death, sorry, because the probability of success drops to 30%, and that's because you want to show the efficacy at this point. So your molecule is safe, but is it efficient in hitting the endpoint that you have for your study? Because you design a study to say my molecule will be efficacious enough if hits this target.
Yes.
Do you hit that target or not? It's a pretty standard question, and there's no way out. You either
that's where you find out. Yeah, no, you can't cheat here.
At that point, phase 3, which are much larger trial, where you want to see if that efficacy is confirmed in a broader population. At that point, the probability of success bumps up again to 65% again. So yeah. And if you multiply 65 by 30 something and by 65 again, that will come that 10% we're talking about.
That you see, yeah, exactly. No, I mean, we all, I mean, the people that were involved, at least as a part of their career in this process, they all know that it's probably one of the, highest failure field of science in terms of
it's unforgiving. It's an unforgiving field, and yeah, sometimes it's frustrating, but it's also very exciting.
It's also very exciting, yeah. And I feel like it is a process where you can see a lot of, I mean, of course, you will have the aim and the mission to, improve patients' life with your drug. So you still have to keep that in mind. But it's also like something that, gives you the opportunity to, work with many different people there, because, and here I want them to talk a little bit more about your job, what you do. So it's, drug discovery and drug development is a very multidisciplinary effort from everyone, because we see like pharmacology people, we see informatics, chemists, we see clinicians that are involved, the people that are expert in formulation. And in your job, you, if I got it right, you are like sort of trying to manage all of these figures into this, common goal of discovering a novel drug. And how do you approach this? I mean, the fact that people have different backgrounds and people have different knowledge. How is that in your daily life? Let's say in work.
That's interesting. That probably the most interesting part of my job, to deal with so many professionals. Like you said, pharmaceutical industry brings together a huge number of different professionals, from medical doctors to pharmacists, chemists, biologists, informaticians, you name it. So it's a very technological space, and that's very important, because yes, it really brings the technology forward in terms of driving also the knowledge of the industrial development in this field. My job sometimes it's not the best job, because I need to make sure, I need to take care of the 3 main constraints that every project has, which are time, cost, and the scope of the project. So you want to make sure you do what you got to do with the money you're given and the time you're given, and even less, because everybody says there's a patient at the end of the line. So the sooner you do, the better it is. So it's not easy sometimes, because, like we said, the failure rate is very high in our field, and specifically I work in the early stages of the pipeline. So lead discovery, lead optimization, candidate selection, preclinical phase, which is where you see a lot of failure. It's estimated that yes, For every now depends really on your specific from your company, your probability of success and your attrition rate. But on average, I calculated that for every approved drug, there are at least 30 projects that failed. And that's what accounts for also the cost of developing a medicine. Because again, if you Google how much does it cost to develop a medicine, you will probably come up with $1 billion. This is the most the figure that comes up most when you dig that. And I think there's a little bit of debunking to do here, because $1 billion is not the actual cost to develop that single medicine. So if you take the cost to go from target validation to the approval of one single medicine, it's not $1 billion, it's less. But you have to account into all the projects that failed to bring that molecule forward, your attrition rate. So if you apply the attrition rate to that money, you will end up that, yes, you probably had 30 projects in lead discovery, and then you brought forward, I don't know, let's say 10 or 15 to the preclinical phase, and then preclinical phase, you've got another 5 or 6 then made to the clinical, and then in phase 2 you only have 2, and then 1 in phase 3 and approval, so on. And that all accounts for the cost of the research.
Yes.
The only thing that we can do to improve the probability of success is manage things in an orderly fashion. That's why research and development are processes that are organized under a precise management system, which we call the pipeline. Which is not a fancy word for, but it's a specific tool of management to make sure that there's a precise strategy for the management of the portfolio. So you make sure you divide the life cycle of a project and all the projects that you have in your industry and in your company into different stages. Like we said, lead discovery, lead optimization, and so on. And you've got stage gates. So milestone, you get there. And at that point, you can evaluate and say, okay, did I satisfy the criteria that I had? Yes or no? Okay, this is moving forward. This is not moving forward. And at that point, you will have a broad vision of your portfolio and say, okay, this year, we're going to have X molecule that will select a candidate. And next year, we're going to have Y molecules that will go into phase 1. And that's how you approach that. And you make sure your pipeline is never dry by those calculations, which are it's not rocket science, right? But it's something that really gives you the sense of where you are and specifically where you will be 10 years from now, because this is the bottom line of the research and development, to project the company into the future. Today, we're successful because we have this product, but what's going to happen in 10 years? Nobody knows, but we need to make sure we do something in order to remain competent in the market, of course, because as any company wants to survive, it's not only a matter of money. I'm sorry to say that, but and of course, you want to make sure you improve through the years, and you have your pipeline again is never dry.
Yeah, exactly. So it is something that would help you at least to try to predict the future. Of course, it's not an exact science. You say it's not rocket science, but I think it's still quite complex, and it's still a lot of hard and tough decision to be made in this process. And I totally understand that you need to have a full let's say, overlook view of what's going on and what are the attrition that you can find, how you keep the process up and running. And I don't know if, you can share anything about that, about your current company, but how the new technology are helping in this process, like to make a better decision faster. You're using any, there is AI now. It's everywhere, and they try to use it as much as they can. But how this can be applied in the management of a drug discovery pipeline like you do?
Yeah. Well, a lot. I'm not an expert, so I won't go into the detail of that because nowadays everybody is an AI expert. But I'm really fascinated by that technology, but I don't know enough to talk about it. I think AI is having an impact on virtually everything, and the pharmaceutical industry is no exception. Like you said, making decision is very important. It is really the ultimate goal. And my job is really to make sure we provide the information required to make decisions and to make good decision possibly. But you never know that. But at least I try to give upper management and other team members and other people the information to make informed decisions. So this is going to cost us this, and we're going to be there then. Do you think it's a good choice to pursue this road? Yes. AI is changing a lot of things. I can speak for my field, so the project management field. Now, pharmaceutical industry is adopting AI, of course. I can speak also for my company. In our company, but I believe in any company, there are policies for the use of artificial intelligence and generative AI tools, because free tools, we know if it's free, like they say, the product is you. Because they want your information, so we are not allowed. And I fully understand this, and I'm very favorable of this. We're not allowed to put information that are specific to our company, so sensible information, into free tools of generative AI, because very simply put, I have a meeting and I need to draft a minute. I cannot use a generative AI tool from the internet to do that, because I may use sensitive information that maybe can be used to I don't know, infringe prior art for a patent by somebody else. You never know. So you don't want to do that. So what we do, GSK, and what I think other companies are doing, we have our own AI tools, which are the client version of generative AI. I cannot speak again related to the drug discovery process, because there's a whole sector of our industry who is working on that, and it's not. I'm not an expert again. I can speak about myself. Mm-hmm. So I did this experiment with our generative tool AI. And project managers, you don't have to. There's no specific degree or something to become a project manager. You just become a project manager. There are certification, of course, that make sure you adhere to some standards, international standards. And the most probably famous is the PMI, the Project Management Institute, with the PMP certification. So what I did is I submitted to our tool 15 questions from the PMP exam, and to see how the AI scored, and this tool scored above target, which is the highest. And I thought, okay, this tool, yes, is a good project manager, because if I submit to the tool a scenario or something that is going on, a risk or something that I need to take into consideration, this tool can give me an insight on how to manage this correctly, probably, if I give the correct prompt and the correct information. So it's great. And here comes another question, I think. So many people are scared that this tool can take their job. And personally, I don't think so. I don't think that the AI will take the job of project managers, but I think that project managers who use AI will take the job of project managers who don't. Yes. Because again, when the internet came out, when Google came out, I mean, look where we are now. Google probably created a gazillion number of jobs, and so will AI. Of course, we also take away some jobs, like the car. When they invented the car, yes. Of course, the blacksmith can no longer put a horseshoe on the horses. But yeah, that's just
this part. Yeah, I mean, of course, all the jobs, I mean, will need to evolve, and we are like, I think, in the optimal position to be, knowledgeable enough to get ready to this big change that is coming. That, yeah, it's somehow like the example that you made, the advent of cars against people using, horses to move around. And that is the kind of breakthrough with AI.
You're right. This is a breakthrough moment.
And we are in that position where we can, adapt our job and evolve our job to, include the AI in our, daily work. And as you said, people that would be totally against it, they will need to probably rethink a little bit their position, because it's going to be part of everyone's life. And this means also that, as you said, there is a lot of fear in terms of how this is going to be implemented, how this will change our life. So it's important on how we communicate these kind of things outside of our works, because people are a little bit afraid. People, when there is this kind of uncertain moments, there is a lot of misinformation, and people get carried away with that in terms of understanding what is true and what is false. And here I want to start also mentioning what you do as well with to help this kind of communication outside of the AI, but outside of the pharma world, to help a good let's say diffusion of our work outside. I'm saying that because from everywhere, like movies and books, the pharma company is always described as the villains. They are like the bad guys.
The bad guys.
And
oh, please go finish.
Yeah. And this is not like, of course, this is not true, but also like the only way we can do it is to help people outside of our work, or what we do, and how we want to work with them and towards the better, outcomes for everyone's lives. And I've read you. I know that you have written a book that I read it, and thank you very much for sharing that with me. It was fascinating, where you collect these, experience from famous scientists that discovered famous like really famous drugs, but also you are working a lot on like social media and like with other initiatives to support this communication. So can you tell us like a little bit more about that? That would be very interesting.
Yeah, like you said, I think it's very important to explain how things work. Because yes, the pharmaceutical industry doesn't have the best reputation in the world, of course. That has to do with many factors, which I don't want to discuss today here. But yeah, the bottom line is, yes, the pharmaceutical industry, especially lately, has been her reputation really plummeted. So my goal is to try to divulge information around how medicine are developed, how pharmaceutical research is carried out, to make sure people really understand our job and can have a better idea, a better sense of how we bring medicines to the market, how these products cannot be perfect, because yes, these are products that interact with a biological entity, with a body, and so the result is always not unpredictable, because yes, we go through very rigorous testing, but yeah, it's never something like, I don't know, a phone that would behave always like that. Anybody who uses that will have the same outcome. So some years ago, I thought, okay, why don't we start to divulge information on LinkedIn, on social media, to make sure, yes, we make people, a broader audience, understand how pharmaceutical research is carried out. And I was surprised by the interest. Of course, the majority of my audience are people from the industry, because yes, I speak things that I say things that resonate with them. But there is also a component of people who look at my posts and follow my activity that is not from the industry, and it's really nice to see that, because at the beginning my goal was to show somehow that even the industrial research can be a social matter, and I kind of did that, because yes, it's something that was not very common on social media. Now it's more common, so I'm very happy to be part of this, and I'm really happy that there's a way to speak about that, maybe in a lighter way, not only under strictly scientific terms. Of course, we need to use a terminology because it's a scientific field, and words matter. But you can always use even light or funny means things to explain things, and people like that. And the book that I wrote was part of this framework, let's call it. So it's a collection of short stories. These are 25 short stories about scientists, the finest men and women who had been working for the pharmaceutical industry, and they've come up inventing medicine that actually changed the history of humankind, because they brought us to antibiotics, aspirin, anesthetics, things that really changed the way medical treatments are applied, and also, yeah, they brought us into the modern era. And again, that was part of a framework to communicate to a public, which of course is specialistic in terms they already speak that language, but also to show how through the development and through the evolution of the field, we've come from, very uncharacterized and almost magical product to an industrial product, which is really satisfy the highest standard of the industry today, and also it's an overview of the human history, because yes, so imagine that less than 100 years ago, you could just go out and have a very minor wound and die of sepsis and die of bacterial infection. There's this episode in the book where Calvin Coolidge Jr., who was the son of the president of the United States, so not a nobody, a poor guy, he was playing with his brother in the courtyard of the White House. They were playing tennis, and he got a blister on his foot. And that blister got infected, and the infection turned into sepsis, and the son of the president of the United States died because of a blister.
Yeah,
so those were the days. And I think understanding the history, the past of how those medicines were discovered and developed really helps us also understand the present, and how this industry works, and how we treat medical issues, and how we discover medicines.
And we are also like evolving as a species as well. Like, exactly 100 years ago, which in the general let's say timeline of humanity, it's like a blink of an eye. It was kind of normal to die out of an infection, out of a blister, or a cut or a wound. And now it feels like we can treat diseases that we didn't even know back then. So, and we can improve, like the quality of life, and improve the length of people's life. And so this is like also a nice story on exactly how through medicine we evolve, let's say, as a species towards better outcomes and better life for everyone.
Quality of life, of course, a better quality of life. Yes,
that
absolutely.
is super important. And I mean, I read the book and I found it very, very fascinating. I mean, it is in Italian, so if you want to tell the title, people would be happy to know more about that.
Yeah, absolutely. It's in Italian, like you said. The title in Italian is Scienziati in Azienda. That can be translated something like company scientists. Let me do some commercial here.
So sure, go ahead.
Easily find on Amazon. It's both a hard copy or an ebook. It's not very expensive. My goal is not to make money out of that, but again, my goal is really to divulge and some culture around pharmaceutical industry and how pharmaceutical products and medicine are discovered and delivered to the people.
Yes, exactly. And that's what I was about to say. Like I read the book and I feel like the language that you use is like very accessible also for people that might have an interest in this field, but they're not specifically in the field. So they want to know more. They want to know a little bit of the history of the people that were part of this big family, let's say, of drug discovery and drug development. So it is a nice way to get also introduced to this world. And here I'm thinking about also like the younger, generation, the people that probably are now studying pharmaceutical chemistry, or they're studying pharmacology or medicine, and they have this idea of, what about moving more towards the drug discovery? What about this field? Of course, as you said, in the last few years, due to the pandemic didn't help, the misinformation and fake news that you find on the internet, the reputation of our field has been a little bit affected by that, I would say. So, going back to the people that are now studying, like to work with them and convince them and tell them about our job and our work would make them probably more willing to pursue a career in our field. And I know you collaborate with someone also on that matter. Maybe if you want to.
Yes, this is part of the that I always like to do. So try to connect with the younger generation to coach them on our industry, and also maybe on how to get a foot into the door. I've been invited a number of times to speak to PhD students or other students, even high school students. Because again, I think it's very important to make people understand how things work. We're an industry like any other industry. We need to abide to some rules. The rules for the pharmaceutical industry are stricter than any other industry on the planet. The food industry is another very regulated industry, but not as much as the pharmaceutical, of course. So it's very important, and I really like to give back something. So yes, even part of the posts that I publish on LinkedIn sometimes talk about this matter. So how to orientate people, students to the industry, because at least in the Italian environment, and this is something I lived firsthand, university does not prepare you to a career outside academia. And this is frustrating, to say the least, because it's clear. I mean, I come from a very big university. The University of Padua has something like 70 or 80,000 students from a town that has a resident population of 250 people. So can you imagine that? And you cannot imagine that the university can actually give a job, or at least foresee a career for every one of them. So I think it would be very, very important to prepare people to the industry, even when you're working at your PhD maybe, or your master thesis, to make sure, of course, there's a job outside, but what kind of job you can do. Like you said, pharmaceutical industry is an industry where there are a huge number of professionals. So, and many people don't know that with their degree, they can work in the pharmaceutical industry, or what can they do in the pharmaceutical industry, medical affairs, regulatory affairs. Many students don't have the palest idea of how a pharmaceutical company works, what are the functions and the job that are into the pharmaceutical company. Like many people working in a pharmacy with a pharmacy degree, they could have a very good career in regulatory affairs. They could have a very good career also in production. To be a qualified person, you need a specific degree in Italy, whether it's 5 years degree, so so-called vecchio ordinamento, old. So when I speak to young people, I realize they don't know this. They really don't know. Nobody tell them. And I say, wow, that's very straightforward. Why nobody told me that?
Yeah.
And this is the question.
Exactly. That's a good question for
that. Why nobody told me when I was a student that there are plenty of opportunities. Of course, it's a very competitive landscape. You don't have to think, okay, I'm going to the pharmaceutical industry and they'll open the door and give me money. Of course, it's tough. The competition is tough, and there's always the old problem that you need experience to get a job, but you don't have experience to get your foot into the door. But nonetheless, if you at least had this information, and this is what I try to spread, you can work toward that, and you can get more information and maybe train towards that and make some experience, or at least look toward that direction.
And what is the suggestion that you would give then to because as you say, the transition between academia, like when you are students, and the company can be quite tough. I felt that transition even when I did my transition from academia to the private sector, and I was a postdoc, so already considered, let's say, a professional. So what kind of suggestion would you give to young people or like a early professional that wants to do this transition from academia to the industry or the private sector in general?
Leverage today's technologies like social media, get in contact with people. I get contacted many times on LinkedIn by people who says, how I saw your profile, I saw you went from the academia to the industry, how you do that? And I really like when I have time I try to answer as much people as I can. So today there are a lot of tools to be in contact with people. So try to really contact people, because the way you find job today is still by contacts. Okay, you can always apply for a position and you can get a job, but yeah, the probability increases if people know you, and that doesn't mean you're a relative or you're the son of somebody, or that means you're somebody who, I don't know, it's been in touch with people, so have shown interest in what they do, and maybe they start publishing something on LinkedIn or other social media, and they become known for that, for their expertise. So again, the simplest piece of advice that I can give is leverage those tools to make sure, yes, you start building your knowledge, and not only of a matter, but also of people in the industry, and try also to exploit as much as you can those tools that university gives you, like stages within an industry, or an industrial PhD, or any other thing that really can makes you, again, put your foot into the door, and gain the little experience that will help you land your first job.
Yeah, no, that's great. That's great bit of advice. Also, because I think there is a need to foster this sense of community among scientists, and start, this kind of collaboration, start this kind of connection. And of course, like we have tools like LinkedIn or other social media that can help you do that. So like, don't be shy and go ahead, start, asking questions.
Worst can happen. People won't reply to you.
Exactly. And it's important, because then exactly you will see then maybe in the future you will find other people that you have been in contact with, and you are, I don't know, 10 years down the line in your career, and you will see that now you have this sort of connection that you probably didn't remember, but it will come in handy later on. Okay, well, I think this is a nice closing piece for our episode. Like,
Agree.
don't be shy, go and talk and create a novel
out.
community of science. Yeah, and reach out.
Well,
So I want to thank you, Carlo, first of all, for being here with me
I
today.
really enjoyed a lot today.
Thank you.
Talking science, talking about our field. Thanks again for inviting me.
No, thank you for being here. It has been a very, very interesting chat we had today. And I thank you, and thank you all for listening. Bye-bye.
Thanks. Bye.